Our first presentation for session 2 comes from Dr. Mike Pacanowski. He's the director of the Division of Translational and Precision Medicine in the Office of Clinical Pharmacology at the FDA. His presentation is titled Precision Medicine 2030.
Good afternoon. My name is Mike Pacanowski, and I direct the Division of Translational and Precision Medicine in CDER's Office of Clinical Pharmacology. I'd like to thank the organizers for the opportunity to be here with you. I will say up front, I am not an expert by any means in AI methodologies, so for this portion of the session, we're going to zoom out a little bit and not talk about specific case examples or technicalities, but rather how AI fits into the general landscape of precision medicine. So we'll spend some time going over the current environment and different contexts where AI could prove potentially useful. To start, it's appropriate to keep in mind where we are in the grand scheme of things. The first evidence of medicine surfaced several thousands of years ago. However, most everything we know of modern medicine really began in the 1800s, and the technologies to treat human disease really diversified quite a bit in just the past couple of decades, as we've seen growth in drug products that are biological, oligonucleotides, cell and gene therapies. The diagnostics have evolved quite a bit as well. We now have high throughput approaches to sequence the human genome in days, whereas it originally took over 13 years to sequence the first draft of the human genome. So the pace of innovation is pretty remarkable. And precision medicine, as we think of it now in modern terms, is a relatively recent enhancement to medical practice that has grown quite a lot in the past 10 to 20 years. However, our ability to match therapeutics to any given individual's disease is still probably in its infancy. And perhaps our current practices will be viewed as crude 10 to 20 years from now. And all of this is to say that I'm really excited for the future and the role that computational power and methodologies like artificial intelligence can potentially play in individualizing therapies. Now, in broad terms, precision medicine basically amounts to tailoring treatments based on individual characteristics. In our current environment, many think of this as approaches based on genomic testing or gene therapies. And we in the Center for Drugs have been focused on targeted therapies, which has many different definitions, but what I commonly think of is using some biomarker test or other type of tool to select patients for treatment, to guide dosing, or to calibrate the monitoring approach. Now, the biomarkers that are used to accomplish this are either used at a point in time to forecast future events, like susceptibility biomarkers, prognostic biomarkers, or predictive biomarkers. Alternatively, they can also provide information relative to the past, as in the case of monitoring biomarkers, response biomarkers, and some safety biomarkers. And this taxonomy of biomarkers is something that was developed by an NIH FDA collaborative effort and is contained as a resource known as BEST. Now, everything physicians do in practice has historically been personalized. And what we think of as modern precision medicine, where we're taking some technology and applying it to guiding treatment, stems from early pharmacogenetics, where studies investigated the role of genetic variations on drug metabolism. This then evolved into studying patients who have specific molecular characteristics. And earliest examples of this were trastuzumab for patients with HER2-positive tumors, which happened in the late 1990s. As our genomic tools have gotten better, we've been able to understand the pathogenesis of many diseases much better, and this has then resulted in reverse engineering of some of these data to help inform the design of new drugs, as was the case for PCSK9 inhibitors. We've also seen expansion of new types of drugs that are genetically targeted, such as the synthetic oligonucleotides that interfere with RNA at the genetic level. And we've had a number of these types of drugs approved for many rare diseases. Now, in more recent times, what we've seen is greater complexity of the types of biomarkers that are being developed and the populations that are being targeted. For example, we've had a number of drugs approved for molecular characteristics rather than a tumor type in a specific location in the body. And we've also seen much more complex biomarkers that have tests for many different genes and variants in those genes. Even more recently, we've had a number of cases of drugs being designed and developed for an individual patient because of their genetic characteristics. And that's an opportunity really afforded by antisense oligonucleotides and the ability to change the sequence of a drug very easily. When a biomarker, or more broadly a tool, is identified, there are a few ways for it to be recognized by FDA. Community consensus on the use of a given tool to help treat patients may result in it being a standard of care that is naturally adopted in clinical trials. In cases where the tool is intended for broader use across multiple drug development programs, as might be the case with outputs from pre-competitive consortia efforts, FDA has created a qualification process to review the development, evidence, and application of the tool, so it doesn't need to be renegotiated every time it is applied in clinical trials. Lastly, FDA also works with individual drug, diagnostic, or other medical product developers on how to implement use of novel tools in clinical trials and eventually practice, and we'll show some examples of that in the next couple of slides. A few representative examples are shown here. This list contains several drugs that were approved in 2022 that have precision medicine elements to them. As is usually the case, we have a number of cancer therapies approved for molecular subsets because the mechanism of the drug will often depend on the presence of certain molecular alterations in the tumor. Now, over the years, as the number of drug approvals has steadily increased, so has the number of drugs that are approved for molecularly defined subsets, or drugs that also have tailored dosing based on some genetic characteristics, or that are mechanistically driven treatments for rare genetic diseases. And precision medicines, quote unquote, occupy roughly about 1 in 5 approvals each year. So that, in a nutshell, is where we're at with respect to the basic landscape of precision medicine. For the next few minutes, I'd like to talk a bit more about areas where AI approaches can fit into the precision medicine paradigm. So I'm a clinical pharmacologist, and our office's staff have a very specific focus in the regulatory review process that is summed up by these 4 basic questions. We are basically concerned with ensuring that the drug meets the substantial evidence standard, be that through primary evidence from clinical trials, potentially including surrogate endpoints, or as through confirmatory evidence in support of a single clinical trial that might be conducted. Dosing is a very major focus of our office, and we place a lot of emphasis on seeing how drug concentrations match with the effects on the disease, as well as how we go about managing the drug in the setting of various factors that can make people vulnerable to adverse events, such as high exposures that stem from drug interactions. So evidence of effectiveness and therapeutic individualization are part and parcel to our day-to-day work. All well aware, drug development is really an exercise in reducing uncertainty about a drug's benefits and risks in the context of treating a particular disease or condition. And what is shown here in a rather busy way is the battery of clinical pharmacology studies typically conducted to describe a drug's pharmacology, its disposition, its efficacy, and its safety. And all of this information allows us to evaluate those benefits and risks and individualize the therapy where needed. And we can take into consideration many factors and evaluate the totality of evidence to answer those pivotal questions I showed on the previous slide. And now, relative to the universe of data collected in the post-market setting that many in the audience may be accustomed to working with, we have a much smaller volume, but also a very rich set of data, and using advanced analytical methods could help integrate these data quite a bit. Now, that collection of studies really helps inform important aspects related to the drug's pharmacology and disposition. But it also helps us understand how the drug works and in whom, and what factors might be influencing those responses. So as specifically related to precision medicine approaches, this slide summarizes some of the main triggers that make a precision medicine approach more relevant, at least from our perspective. So to illustrate, in situations when you have a very broad therapeutic range and the drug is pretty well tolerated, layering a test to guide its use might not have much utility. However, when the drug has a narrow therapeutic index, an unclear mechanism of action, the disease may not be well understood, or it has high degrees of observed variability or heterogeneity in treatment effects, it then becomes prudent to investigate factors that can be used to guide that therapy. And as shown in the earlier slides, most of the current examples of precision medicines have relatively well understood disease pathology and a targeted mechanism of action. And that makes targeted drug development and the attendant improvements in the benefit-risk profile much more feasible to pursue in the drug development context. Now, when little is known, we do have the ability to conduct investigations to help identify those factors that influence response. And data can be drawn from clinical trials or many other sources to help understand disease pathobiology and drug pharmacology. It's possible now to harvest DNA to perform whole genome sequencing, generate data from other biospecimens using other omics techniques, and so on. So the research and discovery side, we have a really tremendous ability to generate tremendous amounts of data on any given patient. And again, this is where it seems quite intuitive to apply AI and machine learning approaches that allow us to take those data and find those mechanisms for differential response or identify the subsets in whom the benefit risk profile may differ. And once identified, the result could then be used for a number of different purposes, such as to aid in assessing risk for outcomes, confirming or refining diagnoses, identifying responders, selecting doses, and so on. And this can be the basis for various clinical trial maneuvers, such as conducting enrichment trial designs or developing diagnostics that could be used to select patients in the clinic. One thing that is critical is that patients have to be tested for various things, and specimens need to be collected that allow this research to be conducted. So some forethought is required, and deeply phenotyping patients should be an essential part of drug development. Now, one difficulty we face is that our approach is still fairly simplistic. In the example shown so far, only a limited number of predictors, like gene variants, might be evaluated based on some strong biological hypothesis, and that result is then reduced to a binary decision, even if the test produces a quantitative result. So shown here is a number of drugs that have been approved for lung tumors in recent years. And as you can see, in virtually every case, there is a single pathogenic driver relevant to the mechanism of the drug that was targeted for drug development. In some cases, it might be many variants that might be targeted, but is still anchored by that single dimension of pathology. However, on the right, you can see that tumors are much more complicated than that. And we don't really integrate all of the other factors that might be present, such as methylation patterns, copy number, and so on. And many aberrations may coexist. If we were able to integrate these data, we could potentially predict responses to the drugs or design a treatment strategy that is much more effective. Now, molecular complexity is just one layer, and there are many other patient factors that can drive response to medicines as well. And warfarin represents a good example of the complex interplay of factors and how that can be reduced to practice in drug labeling. So genetic variants here in the drug target, as well as the drug metabolizing enzyme, are major drivers of response, and thus the dosage requirement. But many other factors come into play. And you can see here an attempt to integrate that information by providing a rubric for the dominant drivers, specifically the genotypes, that can then be augmented by knowledge of clinical factors. Now, it's challenging to address this type of complexity in labeling. As such, if going beyond qualitative approximations of dose, calculators or tools would need to be developed to implement precision dosing at the bedside. And for warfarin, such tools have been developed and are available for use in practice, such as for warfarindosing.org. Now, we at the FDA tend to focus on a single drug. In many practice settings, prescribers have multiple therapeutic options. And this Sankey diagram was pulled from a recent abstract and shows the care patterns for patients undergoing treatment with TKIs for chronic myeloid leukemia. I think most would agree that it would be best to select the drug that is most likely to work first and give it at a dose that's not unacceptably toxic. And here you can see the diversity in initial drug chosen, what they switched to, and ultimately went on for multiple different iterations of this process. And all of this trial and error might be based on response or tolerability. There's a tremendous amount we can learn from this and understanding individual patient care patterns. It might even be possible to define an approach for a patient based on the experience of other similar patients, or better yet, identify those factors that determine response and create tools, whether molecular tests or something else, that could increase the likelihood that the best drug is chosen first. What now is standing in the way of using AI in the advancement of precision medicine? And this not being my area of expertise, I opted to ask ChatGPT, and the result with some modification is basically shown here. And these are all issues, I think, that are common to all real-world clinical research and adoption of new technologies. Major focus areas for the future should include ensuring that diverse data sources can be integrated to improve the quality of models that are generated. However, there are many challenges with how real-world data are assembled, and that is a matter in and of itself. But bottom line is better data, better models, more collaboration to help advance development and implementation. So to wrap up, we are at an interesting stage of medical product development, and perhaps we'll experience some growing pains with the adoption of new technologies. But advanced data analytics are really one facet to the next generation of precision healthcare. And that will be enabled by a number of things: omic technologies, the Internet of Things, complex diagnostics, innovative therapies, and improved data aggregation infrastructure. Now, to conclude, precision medicine approaches really rely on technologies that improve the mechanistic understanding of disease and drug response. AI and complex data analytics can augment the use of those tools and further reduce biological complexity, optimizing drug development. And in practice, this can help resolve multiple factors to support therapeutic decision making. And with that, I'd like to thank you for your attention and turn it over to the next speaker.